Good morning.
Morning.
Yes, if you would, type your first name or your nickname into chat for me. Thank you. Thank you very much. Good to see everybody this morning. Very good. So today during lab, we're going to be finishing up our discussion of carbohydrate catabolism. And we're going to begin looking at fermentation. We have not yet talked about fermentation in lab anyway, so we'll look at a test that's very commonly used to test the ability of a microbe to ferment. Very good. If you remember the last time we were together, a couple days ago, we were talking about carbohydrate catabolism, or the way that microbes can break down, catabolize glucose in order to make ATP. All cells require ATP. It is a molecule that is necessary for life on our planet anyway. So we looked at the idea that some organisms are using aerobic cellular respiration, using oxygen as their final electron acceptor at the bottom of what we call the electron transport chain. Other types of microbial cells also do cellular respiration, but they do it a little differently because they use something other than oxygen as their final electron acceptor. Both types of cellular respiration are very efficient. They make ATP from glucose very efficiently. It's a long, long series of reactions, but it produces lots of ATP. So it's a good way to make ATP if you're a microbial organism or any other kind of organism. Cellular respiration is great. Some microbes use cellular respiration as their primary pathway. That's what they prefer to do to make their ATP. But if their final electron acceptor is not available for some reason, they have the ability to switch to a secondary pathway called fermentation. Fermentation will also make ATP, but it doesn't make it nearly as efficiently. In other words, for an equal amount of glucose, if you put it into fermentation, you're not going to get very much ATP. If you put it into respiration, you're going to get a lot of ATP. That's what we mean when we talk about efficiency of these processes. Still, other microbes that we've talked about in here don't really have a need for a primary pathway, but have the ability to switch equally well between respiration and fermentation. And still other microbes only use fermentation. They never use respiration. So we see the whole gamut in microbial life. We see all combinations of which pathways they use, which pathways they prefer, which pathways they don't prefer. We see every possible mix. We talked in here about oxygen requirement categories, if you recall, and thioglycolate medium, how we use that to determine an organism's relationship with oxygen. But we said that test and those designations have nothing to do with whether or not an organism can ferment. So if we want to investigate fermentation, we have to do some additional tests. And we'll start talking about that today. So the last time we were together, we looked at starch hydrolysis, and we looked at oxidative fermentative test, or OF test. And when we finished up last time, we were just beginning an experiment. We were going to create some OF tubes for 3 different organisms. So let's jump back into the lab right where we stopped last time. If you recall, we said that the OF test is a test that is run in pairs. So we have test tubes filled with OF medium, and we always run 2 for an organism. We run 1 tube that has an exposure to air, and we run a second tube that has a little bit of mineral oil layered on top of the medium to help keep the amount of oxygen in the medium relatively low. Always run in pairs. We said the medium starts with a green color, and in the open tube and the closed tube, we're looking for the production of acid, because there's an indicator dye in here that will turn yellow when the pH becomes acidic. So if I see 2 yellow tubes in an OF test, I know that this is an organism that can produce an acid through cellular respiration and through fermentation. If I see a combination, 1 tube yellow, 1 tube green, and it could be either tube for either color, I know that they're capable of one pathway, but not the other. And finally, if both tubes stay green, we know that this organism was not able to use glucose in these conditions. It's important to remember that the OF test is testing for aerobic cellular respiration. So it is possible that we have an organism that is an anaerobe or is very fastidious and just can't grow in OF medium. So we have to be aware of the fact that just because we get 2 green tubes at the end, that doesn't mean we have found an organism that can't use glucose at all. It means that they can't use glucose under these conditions. Remember, all living things on our planet use glucose for energy production, for the production of ATP. So if they don't use the glucose in an OF test, it's not because they can't, it's because they can't use it under these conditions. There's something wrong for them. Maybe it's the presence of oxygen, maybe it's the lack of a particular nutrient. Whatever it is, it has prevented them from using the glucose in the tube. The last thing we talked about was the fact that sometimes we'll pull an OF tube out of the incubator and it will have turned blue. Remember, the indicator dye in this test, and it's called bromthymol blue, it turns yellow in acidic conditions, but it also turns blue in alkaline conditions. Sometimes we get an organism in an OF that is able to use the glucose, but again, can't under these conditions. And so instead, it uses the peptone in OF medium. So that's an organism that's able to grow in OF. So it's not fastidious, in other words. It can grow, it just can't use the glucose under those conditions. And when that happens, it'll turn to the peptone. It will use the peptone in order to survive, and it'll turn the media blue. So 2 green tubes, we've got an organism that can't use glucose under these conditions. If we see a blue tube, we know that organism has turned to the peptone in order to survive, because again, for whatever reason, it can't use the glucose under these conditions. All right. So for our experiment today, we said we were going to use 3 organisms: E. coli, Bacillus subtilis, and Pseudomonas aeruginosa. And we've seen all 3 of these in previous labs. We have 2 gram-negative rods and 1 gram-positive rod today that we're working with. When we finished up last time, we said that we already know something about one of these organisms from one of our experiments. We know that E. coli is a facultative anaerobe. And when we finished up, I had asked the class if you could make any predictions about what E. coli might do in an OF test. We know it's a facultative anaerobe. And if you remember from our discussion of the oxygen categories, facultative anaerobes are very adaptive. They prefer to be in oxygen so they can do aerobic cellular respiration. So if there's oxygen around, they're going to do aerobic cellular respiration. But if there is no oxygen around, it's no big deal for them because they can also do anaerobic cellular respiration as long as the final electron acceptor is available for them. Remember, that's nitrate, sulfate, or carbonate, depending on the species. And if they can't do anaerobic cellular respiration, well, they still can change. They can change and switch pathways and do fermentation instead. So my question to you when we finished up last time was what you expected to see when we inoculate a pair of OF tubes with E. coli. We can't really make predictions yet about the Bacillus subtilis or the Pseudomonas because we don't really know what oxygen category they belong to. But we know that E. coli is a facultative anaerobe. So if I put some E. coli cells into the open air OF tube, and I put some E. coli cells into the oil-covered OF tube, and I put those tubes in the incubator overnight, the question is, what do you expect to see in the morning? Do you expect one or both of those tubes to have turned yellow? Do you expect that they will stay green? Do you expect maybe to see them blue, possibly? We have to think about what E. coli is capable of. OF medium is a basal medium with a lot of glucose added to it, carbohydrate added to it. There are no anaerobic final electron acceptors in OF. But there's oxygen in that open air tube. What do you think? Brooke and Brianna have jumped in. Good job. You both got it. Think about the open air tube first. The OF is going to have some oxygen in it because that tube is open to the air. And we know that E. coli is an organism that prefers to do aerobic cellular respiration. There's a lot of glucose in that tube. E. coli is not fastidious. E. coli is going to grow in OF, in other words. It doesn't have any problems growing in OF. And since there's glucose and oxygen available, it's going to do aerobic cellular respiration and break down that glucose all the way to carbon dioxide. The carbon dioxide then is going to mix with water and become carbonic acid, and that tube is going to turn yellow. Remember, all the electrons that come out of the glucose molecule that's been broken down, all of those electrons are going to be sent down the electron transport chain so that their energy can be harvested to make ATP. At the bottom of the electron transport chain, there has to be a final electron acceptor molecule, or it can't work. In this case, it's going to be oxygen. So E. coli will happily grow and do cellular respiration in that open air tube. But it will also do it in a closed tube, in the oil-covered tube, because E. coli can happily ferment too. E. coli is happy to sit in the oil-covered tube and ferment. It's going to take the glucose down a different pathway. It's going to take the glucose, it's going to begin to break it down because fermentation involves the first part of cellular respiration, which is a process called glycolysis. Glycolysis is a 10-step reaction. It takes glucose and it turns it into pyruvate. Normally in respiration, the pyruvate keeps getting broken down. But in fermentation, instead, the pyruvate gets sent down another pathway and it gets turned into one of three things, or I should say, combinations of alcohol, acid, and gas, depending on the species. Fermentation always produces some kind of combination of alcohol, acid, and gas. And an acid is always involved. So when E. coli ferments glucose, it creates an acid, lactic acid, as a matter of fact, and the tube turns yellow. Very adaptive organism, E. coli. Good job, Brooke and Brianna. All right, let's take a look at the other organisms as well. Just a reminder that OF medium is a basal medium. So it is possible that you'll put a microbe in that tube that cannot grow, period, let alone use the glucose. It just can't grow. If it's fastidious and it has weird nutritional requirements, it's not going to grow. So it's important to remember that. It's also important to remember that there is nothing in OF medium that allows an organism to do anaerobic cellular respiration. There are no final electron acceptors other than oxygen in that tube. There's no nitrate, there's no sulfate, there's no carbonate. So we're only testing for aerobic respiration and or fermentation. So we already looked at the starch hydrolysis test, remember, when we worked together last time, and we saw that this Bacillus subtilis organism had a positive result. Very interesting. Bacillus subtilis was able to secrete an exoenzyme called amylase into this starch agar and break it down into glucose. That clear area around the streak of cells no longer has starch in it, because if it did, it would have turned black when the iodine was added. Instead, it's this sort of golden clear color. The other two organisms we're looking at today, they don't secrete amylase because the starch didn't change for them. Here are our other organisms in their OF tubes. Here's E. coli. And you could see that both tubes turn this nice bright yellow. As I said last time, the nice thing about OF testing is that there is no question if the tube turns yellow. It's not a shy yellow color. It's very bright. Now, take a look at the other two. Let's start with Bacillus over here. The open air Bacillus tube has started to turn blue. It's not blue all the way down, but there is definitely some blue in here, which means the pH of this tube is alkaline. And that means that this organism, rather than using the glucose to make ATP, has instead been breaking down the peptone. Remember, living things on this planet use glucose and other carbohydrates for energy. And when I say for energy, I mean for ATP production. That's what they're using the glucose for. They're using it to make ATP. So that bacillus right now is struggling because it can't use the carbohydrate in the tube to make its ATP. What it has to do in that case is take that peptone, break it apart, and build its own carbohydrate so it can then use the carbohydrate to make ATP. This is not an ideal situation. Protein is not used to make ATP unless there's no other survival for that organism. A cell will always use carbohydrate if it has it available. It's just that this test has a limitation, right? It's only got glucose available in an aerobic condition. So what that bacillus is experiencing in that open air tube is an inability to do aerobic cellular respiration. That tells us something about that organism. It's still growing. How do I know it's growing? Because it's metabolizing the peptone. So it's not fastidious. It's growing in there, but it can't do aerobic cellular respiration. So what does a bacterium do? It doesn't want to die. So it's going to take that peptone, it's going to break it apart to get the carbon out of it, and it's going to build some carbohydrate for itself that it can then use to make ATP. It's a bad way to make ATP, and the organism probably can't do it for too long. That's the other thing to know. When an organism switches to pure protein, it doesn't have a whole long time to live. Make sense? So the bacillus is alive in there, but it can't do aerobic cellular respiration. We've learned something about it. We've learned that it can't do one of those 3 pathways that we've been talking about. Now let's look at the other bacillus tube. This one is green. The tube with the oil over it has stayed green. Now what that tells us is that it's not growing in here. This is a tube where the bacillus has not been able to grow. It can't grow because it can't survive without making ATP. And in a condition where there is no oxygen or very little oxygen, it can't even use the peptone. We don't talk about the specific pathway that this organism uses to break down protein and build carbohydrate. We don't talk about that in this course. You would talk about that in a biochemistry course, but not in this course. We know, though, that if that tube stays green and it doesn't turn yellow or blue, we know that that organism is not growing, it's not metabolizing in there. So in a condition where there's very little oxygen, it's not fermenting. It can't ferment. It's just not one of the things that Bacillus subtilis can do. It can't ferment glucose. That's a green tube. That is no acid produced, which means fermentation is not going on in that tube. Now take a look at the Pseudomonas, because the Pseudomonas is an interesting looking tube. I didn't show you a tube like this, did I? Look at the open air tube. When we first looked at these tubes, we looked at this picture. 2 yellow, 1 green, 2 green. There's nothing over here that shows half yellow, half green. And yet that's exactly what has happened in the Pseudomonas open air tube. This is actually a very classic result for Pseudomonas. These tubes were inoculated with Pseudomonas cells. Then they were put into the incubator for 24 hours. And when we took the tube out, we saw this. There's acid in this tube, but only a little bit, only up close to the top where oxygen is very plentiful. The whole tube didn't turn yellow, only part of the tube turned yellow. So what's going on? This is an indication that the Pseudomonas is growing slowly. That's all it is. Pseudomonas turns out to be a slow grower in a basal medium. It just takes longer for the cells to increase in number. So it's alive and it's dividing, but it's dividing slowly. In other words, it has a long generation time right now. And it is using the glucose for aerobic cellular respiration because that's what is producing the acid and turning the yellow color. It just hasn't done much of it yet because there aren't that many cells in there yet. This is a classic thing for Pseudomonas. So if I pull a tube out of the incubator and it's half yellow or a quarter yellow and the rest is green, how can I confirm that? How can I confirm to myself that this cell is just growing slowly and that if I let it grow a little bit longer, the whole tube will turn yellow? Well, I could put it back in the incubator. I can let it grow more. And that's exactly what we do. We just don't take Pseudomonas out in 24 hours. We let Pseudomonas cook in the incubator for a couple of days. It's just a slow grower in this medium. But if I come back in a few days, that tube's going to be all yellow because that's an organism that can do aerobic cellular respiration. Now, what about the fermentation tube? All green. Pseudomonas cannot ferment glucose. So what did we end up with? We ended up with aerobic cellular respiration for E. coli, aerobic cellular respiration for Pseudomonas, no aerobic cellular respiration for Bacillus subtilis. Fermentation for E. coli, no fermentation for Pseudomonas, no fermentation for Bacillus subtilis. Just to show you this odd feature of Pseudomonas, take a look at this slide. All 6 of these tubes have Pseudomonas in them. The pair of tubes that's labeled A was left in the incubator for 24 hours. So it looks a lot like our 2 tubes. A portion of the open tube has turned yellow, but the rest is still green. This second set of tubes was left for another day, so 48 hours. And as you can see, now the yellow has come down about halfway. And finally, after 5 days in the incubator, the whole tube is yellow. You have to remember that OF is often used as a semi-solid. So there's a little agar in that tube. That's why it's possible for acid to be produced in one area of the tube and not just diffuse down through the rest of the tube. The acid stays up at the top like that because there's a little agar in there and it's hard for the acid product to get all the way through the rest of the tube. So you have to wait until more and more acid gets produced and more of the tube turns yellow. It's just a feature of that organism that when it's in OF, it grows very slowly. Obviously doesn't love OF medium. It obviously doesn't grow great. It doesn't have everything it needs to grow fast in OF. So 3 different organisms, 3 different organisms, 3 very different OF tests. This is another thing that we can do in the laboratory to investigate a bacterium, whether it comes from an environmental sample or a patient sample. We are often faced with identifying a bacterium in the microbiology lab, and it's our job to use different types of culture media to gather data about that organism. We can then take those results from several of these tests and go into reference tables, textbooks, and online sources. And we can look for the organism based on the test results. We can identify the organism based on the test results. In a couple of weeks, when we're finished talking about some of the most common tests that we use in the micro lab, I'm going to give you a set of data about an unknown microbe. I'm going to tell you where this microbe came from, where it was collected, whether it came from the environment or whether it came from a person's body. And I'm going to give you a bunch of test results. I'm going to show you what it did when it was spread on an MSA plate. And I'm going to show you what it did when it was spread on an EMB plate. I'm going to show you what happened when we put it into thioglycolate broth and what happened when we put it into OF medium. You're going to get a lot of results, and it's going to be your job to use some reference tables that I'll provide for you to identify that bacterium and figure out what it is. All right. So remember, when you inoculate a pair of OF tubes, put them in the incubator, leave them overnight, and then pull them out the next day, you're going to do 2 different things in order to figure out what results you have. The first thing you're going to do is evaluate the color. The second thing you're going to do is compare the open and the closed tubes. With regards to color, if the color is green, it means there's been no change at all in that medium. No metabolism of glucose using aerobic cellular respiration. Sorry, I shouldn't talk about the open versus the closed yet. No metabolism of glucose, no metabolism of peptone. That organism is not growing in there. It's not metabolizing. It's not surviving. If the tube turns yellow, we know there's an acidic environment in there. And that means that the glucose in the tube has been catabolized somehow. It's been broken down and produced an acidic product. If the tube turns blue, we know that it's alkaline in there. There's been no metabolism of the glucose, but the peptone has been metabolized in an effort by those cells just to survive. When we compare the open and the closed tube, the open tube is telling us whether or not that organism is doing oxidative catabolism of glucose. In other words, aerobic cellular respiration. In the oil-covered tube, we're learning whether or not that organism is doing fermentative catabolism of glucose. Look at the color, compare the tubes. We have 3 organisms that gave us 3 very different results. I've got acid in both of the E. coli tubes. I've got acid in one of the Pseudomonas tubes, but not the other. I've got alkaline in one of the Bacillus tubes, but not the other. Glucose is being used for cellular respiration, used for fermentation for E. coli. Glucose is being used for aerobic cellular respiration, but not fermentation for Pseudomonas. The glucose is not being used by Bacillus subtilis, but it is showing us that it's breaking down the peptone in the open air tube so it can survive. So the questions that you should be able to answer now, and you're going to see some of these on your homework, is which of those organisms can secrete amylase? Remember the starch plates. Which ones can oxidize or break down glucose using aerobic cellular respiration? Which ones can ferment glucose? And which ones can't do either? Because for whatever reason, under these conditions, they couldn't. Maybe they're doing anaerobic cellular respiration, or maybe they're fastidious. Only one of our organisms was able to break the starch down. Remember, if I took a little bit of that clear agar, if I scooped a little bit of that out and tested it, there would be no starch in here, but there would be lots of glucose, because that's all starch is made out of. It's just glucose all bound together. So the bacillus has broken down the starch into glucose, and the cells will then start absorbing that glucose so they can use it to make ATP. All right. Questions? Any questions? The OF test tests for an organism's ability to ferment, but it's very specific, isn't it? It tests whether or not an organism can ferment glucose. There are lots of organisms that ferment, but it turns out that one of the ways we can identify an organism is by looking at which carbohydrates it can ferment. Okay, it can ferment glucose. That's an important thing to know. But what about sucrose? What about lactose? What about mannitol? There are lots of carbohydrates, and different microbes have the ability to ferment different carbohydrates. Remember, we said all cells use glucose to make ATP in one way or another. But we also talked about how there are carbohydrates that have more than one sugar in them. Remember, we said the carbohydrate called lactose is made out of one glucose and one galactose. So if I was a microbe that could break apart lactose, I could get at the glucose, couldn't I? Maltose is two glucose molecules bound together. If I was an organism that had an enzyme that could break down or catabolize maltose, I could get at that glucose. So we have to also look at which carbohydrates an organism can ferment. The OF test is going to tell us if they can ferment. But now we need a test to show us what they can ferment. And that's what we're going to talk about next with regards to fermentation. There's a test we can run that we can customize. We can put any carbohydrate we want in this test, and we can look to see whether or not the organism is able to break that sugar down and then ferment the glucose. And that's what we'll talk about next. So that's what you should be seeing on your screen now, is the first slide for the fermentation exercise. The first thing we need to do is just put a definition on this word fermentation. And I talk about this in the lecture this week as well. What fermentation is an anaerobic process. In other words, it does not require oxygen. An anaerobic process that converts the product of glycolysis, which is pyruvate, into some combination of alcohol, acid, and or gas. So let's make sure we all understand this statement. Even an organism like you and I that does aerobic cellular respiration, you have to remember that the oxygen isn't required until the end, right? Glycolysis is the first part of cellular respiration. Then comes pyruvate processing. Then comes Krebs cycle. By the end of the Krebs cycle, all that's left of the glucose is carbon dioxide and a lot of electrons. The electrons get fed into the electron transport chain. We get ATP made. In cellular respiration, the first part, the glycolysis part, you don't need oxygen to make that happen. You don't need oxygen till the end of the electron transport chain. So that means even an organism that is an obligate aerobe can do glycolysis if there's no oxygen around. That's all it's going to be able to do. It's not going to be able to finish and make ATP, but it can do glycolysis. It can take glucose and through about 10 steps, it can make pyruvate. If oxygen's around, the pyruvate's going to go down the cellular respiration pathway. If oxygen's not around and the organism is able to, it'll take the pyruvate and it will ferment instead. So glycolysis is the first part, if you will, of fermentation. It's also the first part of cellular respiration. It's that pyruvate molecule that's key. In cellular respiration, pyruvate is going to go through a process called pyruvate processing. That's easy to remember. And then into the Krebs cycle. In fermentation, the pyruvate is going to be converted into some combination of these products. So that's our definition of fermentation. The other thing we need to understand is why. Why would a microbe do this pathway? Now, remember we said it's all about ATP, really. Organisms must have ATP. All organisms must have ATP. So, of course, a big part of the reason that an organism would ferment is because it's trying to get a little ATP. But there's another reason that organisms ferment, and that's to renew one of the molecules that we're going to need to do respiration. Look at this list. This list describes for us the energy reasons behind fermentation. Sometimes an organism is going to ferment to get its ATP when it can't do cellular respiration. So its final electron acceptor molecule is not available right now, so it switches over and uses fermentation to make ATP until it's able to go back and do cellular respiration. This is what we would call a secondary metabolic pathway. Some organisms, though, do cellular respiration and fermentation equally well. Fermentation is not, a backup. Fermentation is a second primary pathway. They can do either one. And finally, there are some microbes that don't do cellular respiration. It seems crazy because it's so effective and efficient. But there are some microbes that only use fermentation. Fermentation is their primary pathway for making ATP. The key thing to remember is that when it comes to respiration and the production of ATP, and remember, we call that second part oxidative phosphorylation. Remember, oxidative does not mean oxygen. It means oxidation reduction reactions. Cellular respiration breaks glucose down to carbon dioxide and a whole bunch of electrons. The electrons then go down the electron transport chain, get accepted by the final electron acceptor, and all of their energy is used to build ATP. That last part, the building of the ATP, is done through oxidative phosphorylation. Some microbes do this aerobically, some do it anaerobically. It can be done with oxygen as the final acceptor. Those are the aerobes. It can be done without oxygen if there's another final electron acceptor. And remember, we have a list of the most common: nitrate, sulfate, carbonate. Those are the anaerobes. Anaerobic cellular respiration is not the same thing as fermentation. I've said it before, I'm going to say it again. Anaerobic cellular respiration is not the same thing as fermentation. Anaerobic cellular respiration is just like aerobic cellular respiration, except a different molecule sits at the bottom of the electron transport chain, something other than oxygen. Fermentation is different. Fermentation diverts one of the products of glycolysis, which is called pyruvate, into a completely different pathway and turns it into either acid, alcohol, and or gas. All cells can oxidize, catabolize, break apart glucose, but the pathways they're doing it are different. And as I said a minute ago, some microbes can use other carbohydrates to get glucose. So they start with a different carbohydrate and they break that down to get the glucose and then go through the breakdown process of glucose. You and I are pretty simple in our metabolic abilities. Microbes are very complex. So we do all these tests because it can help us identify what kind of microbe we have. You and I, we do aerobic cellular respiration to make our ATP. We do limited amounts of lactic acid fermentation. That's it. If you look across all of the bacteria, you can find all kinds of combinations of what they're able to do. So we want to be able to test, can it do aerobic cellular respiration? Can it do anaerobic cellular respiration? What is the electron acceptor it's using at the bottom of the electron transport chain? We haven't talked about that yet. Can it ferment? What can it ferment? Which carbohydrates can it ferment? Remember when we talked about E. coli a couple of weeks ago, we said E. coli is one of the coliforms. And we said a coliform is an intestinal bacterium, or a fecal bacterium, as we call them, that can ferment lactose. That's what defines a coliform. So we find out all of these abilities about a microbe, and then we use that information to identify that microbe. I wanted to point out that, I have a little spelling error here. I wanted to point out that I've put a document up on Blackboard for you. It's a supplementary document. You don't have to read it if you don't want to, if you're not interested. But the other thing microbes are diverse in is how they do glycolysis. You and I can only do glycolysis one way. It's a 10-step process. There are 10 enzymes involved. Our cells take glucose and break it into pyruvate. Some microbes do it the same way we do it. Some microbes do it differently. So I put a little supplement together if you're interested in seeing other forms of glycolysis, because it tends to show up on graduate exams, questions about alternate forms of glycolysis tend to show up on graduate exams. So you might want to keep this little supplement. Anybody who's thinking about maybe going to PA school, or maybe thinking about going to medical school or dental school, I know some of you fall into that category. When the time comes for you to take your graduate preparation exams, you might want to look over that material. The MCATs are notorious for having at least one question about alternate forms of glycolysis. So just a little something extra on Blackboard for you. All right, so respiration, long series of reactions that fall into these three groups. Glycolysis comes first, pyruvate processing comes second, Krebs cycle comes third. During glycolysis, glucose is broken down or oxidized or catabolized into pyruvate. This process doesn't require oxygen, even in aerobes. And during fermentation, glycolysis still takes place. Glucose goes to pyruvate, and the pyruvate is then used to make one of the three products. Fermentation makes ATP, but it makes a small amount relative to respiration. In other words, it's inefficient. For the same amount of glucose, you're going to get less ATP made through fermentation. It also does it through a different mechanism. We talk about this in lecture. Cellular respiration produces ATP through oxidative phosphorylation. In fermentation, the ATP is made through substrate level phosphorylation. I have a note here too that it also regenerates electron carriers, so that once the organism is able to do respiration again, if it can do respiration, it's going to be ready. It's going to have all the electron carriers it needs. Fermentation is very inefficient, but it makes ATP. So it's a commonly used secondary metabolic pathway. And in some microbes, it is the only pathway. I put this diagram in just to remind us how substrate level phosphorylation works. This green monstrous looking thing represents an enzyme, and the areas that are cut out represent the active sites. You can see just barely a white molecule right here with a yellow circle attached to it. That represents a phosphate group. So during substrate level phosphorylation, this enzyme is going to take this substrate and it's going to turn it into a product. And along the way, it's going to take the phosphate group off of the substrate and add it to ATP, add it to ADP, so that ATP is also made. The production of ATP this way is piggybacked on top of making this white molecule product. So the enzyme makes this molecule of white material, whatever it happens to be, and also happens to generate a molecule of ATP along the way. It's called substrate, because here's the substrate, level phosphorylation, because the phosphate group is going to come off of the substrate molecule and go on to ADP, creating ATP. It's an inefficient way to make ATP, but it works. All microbes, all cells can use glucose to make ATP, but many microbial cells anyway can use other sugars to get that glucose They can use lactose, they can use sucrose, some can use fructose, some can use mannitol. It goes on and on. One thing to note is that these other carbohydrates, they're not big polysaccharides, are they? They're either other monosaccharides or they're disaccharides. So the organism has to have the ability to break apart this other carbohydrate to get glucose out of it. You have to have the enzyme in order to do that. So we can test organisms to see if they can break or ferment these other sugars to get to the glucose that they can then send through their fermentation pathway. There are lots of fermentation pathways. And if you look in your textbook, you can find a nice chart that shows all the different fermentation pathways, at least that we know about and make use of. Because remember, a lot of the products that are produced by microbes through fermentation are useful to us. So we purposefully grow bacteria and other microbes like yeast in these commercial facilities. We push these organisms into fermentation, and then we harvest the products they make, these acids and alcohols and gases. We use it for our purposes. So there are lots of fermentation pathways, but there are two most common ones. So most commonly, we see either lactic acid fermentation, that's the one you and I can do, and ethanol fermentation. That's the one yeast are able to do. Lactic acid fermentation uses glucose to make pyruvate. Remember, that's glycolysis. And then the pyruvate is used to make lactic acid and ATP. Ethanol fermentation is a little more complicated, but not by much. Glucose, again, is sent through glycolysis to make pyruvate. The pyruvate is first converted to a molecule called acetaldehyde and a little bit of carbon dioxide. And then the acetaldehyde is converted into ethanol and ATP. Here's just a partial list of some of the products that we harvest commercially from microbes doing fermentation. Ethanol, we mentioned, butanol, butanediol, those are all alcohols. Lactic acid, butyric acid, formic acid, succinic acid, propionic acid. You know, all those acids that you're able to buy commercially for chemistry-related purposes, they all have to come from somewhere. And a lot of times they're being made very naturally by microbial cells. And finally, the two most common gases that are produced through fermentation are carbon dioxide and hydrogen gas. So there are large industries whose sole purpose is to produce fermentation products for sale. And those kind of industries, they hire students who know how to do microbiology, who understand microbiology. Those are not bad jobs. You can work for companies that in their facility have these giant vats full of culture medium, and they culture microbes just continually. And they put them into an environmental situation that forces them to ferment. And then from time to time, they drain out the culture medium and then they distill the product that you're trying to get. If you go into microbreweries, for example, some of the microbreweries will show customers their fermentation vats, where the yeast is. And they put the yeast in culture medium and they feed them things like hops and other sugars, yum. And the product that they're going to harvest is the ethanol, the alcohol the beer or whatever other kind of alcohol they're producing. I had a student several years ago, in fact, who graduated from Keene State College, who went on to get a great job at a big brewery. And his job was to do some quality control around these fermentation vats. So there are jobs out there in microbiology that can be a lot of fun. This is the table that comes right out of your textbook that shows you just some examples of different fermentation pathways, what the end products are, the acids, alcohols, and gases, what microbes are capable of doing these pathways, and some of the commercially viable products that come out of those fermentation products. Lots of really good, useful products, including a lot of food products, are produced through fermentation. So before we jump into our experiment, does anybody have any questions? It's important that you know why the microbe does fermentation versus why we want the microbe to do fermentation. We want the microbe to do it because we want to gather the product. But the microbe does it for two reasons, really. Number one, to make ATP. Number two, to regenerate those electron carrier molecules that I mentioned. So there's two reasons that a microbe would want to do fermentation or would need to do fermentation. I shouldn't say want because they aren't really capable of wanting, but there are two reasons that that cell is doing fermentation. And the primary reason is to make ATP. All right, we don't have a whole lot of time left, but I'm going to at least get started on our experiment today to look at these fermentation tests. And whatever we don't get to today, we will continue with when we get together on Friday. Now, we have three media we're going to look at today. One is called Carbohydrate Fermentation Test. That's a great name, right? Because it tells you right away what the test is about. What the carbohydrate fermentation test is going to allow us to do is to determine if a particular microbe can ferment a specific sugar. So you can put glucose in there. Essentially, what you'd be doing then is making a test very similar to that oil-covered OF tube. But you can also put any other simple sugar into that tube that you're interested in testing for. The other thing that the carbohydrate fermentation test does for us, in addition to determining if the microbe can ferment that sugar, is if a gas is produced. And I'll show you how we know that. The second test has a terrible name. It's called an Mrvp test. This stands for Methyl Red Voges Proskauer test. Now you know why everybody calls it an Mrvp test. Methyl red is the name of an indicator dye. Voges Proskauer is the name of the scientists who invented this test. Now, what this test is going to do for us is it's going to evaluate the acid that's being produced during fermentation. So it's going to help us determine what kind of acid is being produced. And it does that by pH. So the Mrvp test is going to tell us if the acid has a pH less than 4.4, if the acid has a pH between 4.4 and 6, and if the acid has a pH greater than 6. There's even a part of this test that lets us know if a microbe is able to take the acid product and convert it into a molecule called acetoin. And I'll talk about that more in just a few minutes. Finally, we're going to look at a test called the Citrate Test. Now, this is an interesting test. This test is going to test whether or not a microbe can ferment a molecule called citrate specifically. And we're going to be able to see if it can ferment citrate because we're going to get a nice color change in the media. The important thing to know about the citrate test is this. Citrate is not a carbohydrate, but it is a metabolite that is produced during several different metabolic pathways, including the Krebs cycle. And again, whether or not a microbe can ferment citrate tells us a certain characteristic about an organism. So depending on the results of a citrate test, we have data that we can use to identify an organism. Some of them can ferment it, some of them can't. But it is interesting that it's not a carbohydrate. It can be fermented though. So it's a different kind of test. So 3 tests for this exercise. Here's our list of organisms. It's a little longer than what we normally have. We have 5 organisms here. We have our old friend E. coli. We have Enterobacter aerogenes. This is another coliform, and it's a gram-negative rod, just like E. coli. We have an organism called Klebsiella pneumoniae, a gram-negative, rod-shaped, fecal coliform. Klebsiella is an interesting organism because it can be pathogenic. Not all Klebsiella pneumonia is pathogenic, but there are some strains that are very dangerous pathogens. They cause bad diseases like meningitis, pneumonia, like the name suggests, and even a skin condition called cellulitis. Very interesting pathogen. We're going to look at 2 other gram-negative rods that are fecal organisms, intestinal organisms, but they're not coliforms. One of them is called Salmonella sonnei, and one is our old friend Pseudomonas aeruginosa. Now I have 2 names here, which seems confusing. That's because Salmonella sonnei goes through name changes every once in a while. It's one of those organisms that some people want to refer to as a salmonella because they think it evolutionarily is a salmonella, but other scientists think it's better described as a Shigella. So I'm not kidding you. It has gone through probably 4 name changes back and forth between Shigella and salmonella just during my career. So I've got both names listed here because in different sources, different textbooks and different journal articles, some scientists call it Salmonella sonnei, and some call it Shigella sonnei. The important thing to know is that it's an intestinal organism, but it's not a coliform, so it cannot ferment lactose. These are also pathogenic organisms. One of the key features of this organism that can make it pathogenic is this. It's capable of actually getting inside our cells. So it can literally invade our cells. Very scary. And of course, Pseudomonas, we've seen before. Pseudomonas can be perfectly harmless, but it can also be a pathogen. All right, we've only got a minute left today, so I'll finish up with this slide and then I'll let you go. This is what the test media looks like for the carbohydrate fermentation test. These are what the tubes look like when you get them. This is a liquid broth. It has a nice pinky red color to it when it is uninoculated. Remember, I said we're gonna be able to put different carbohydrates in this broth depending on which carbohydrate we're trying to test. Remember too, I said that this is a test that can show us whether or not an organism produces gas through fermentation. And the way that it's done is very clever. Inside this test tube full of liquid, there is a smaller test tube, a little tiny glass test tube called a Durham tube. And what we do is we take the Durham tube and we drop it down into this other test tube upside down. So there's a little glass test tube in here that is upside down. And the idea is when we inoculate this broth, if the organism starts fermenting and producing a gas, some of that gas is going to get trapped inside that upside down Durham tube. If I enlarge this picture here, you can see the Durham tube. It's just a little glass tube that you put inside this other glass tube, but you put it upside down. So when this is full of liquid, and you put bacteria in here and you give them the time to ferment the sugar, if they're making a gas like carbon dioxide or hydrogen gas, some of it is just going to bubble up and escape the tube, but some of it is going to get trapped in here. And what will happen is this tube will float up to the top and you'll see a little gas bubble in it when you get a positive result. Now, the indicator dye that's used in the carbohydrate test is a little bit different than what we've seen so far. The indicator dye is called Phenol Red. Now, this is another indicator that will turn yellow in the presence of acid, so that's not new. We're going to inoculate these tubes. We're going to put them in the incubator for 24 hours, and then we're going to see what happens. Do we get a yellow color change? And do we find gas trapped in the Durham tube? So that's where we'll leave it for today. And when we get together on Friday, we'll take a look at the results and how this particular carbohydrate fermentation test can give us information about those 5 organisms that I listed. We'll also look at the Mrvp test, and we'll look at the citrate test. All right. If anybody has any questions, feel free to put them in the chat. If you have no questions, enjoy the rest of your day, and I will see you on Friday.
Have a good day.
You too.
Professor.
Yes.
I had a question about the lab practical. I needed to take it again for some reason.
Oh, is this Sean?
Yeah.
Yeah, Sean, the exam that I got from you. Oh, and if you're not Sean, you're free to go. I don't mean to hold you up.
I want to say something. So if you want,
oh yeah, you can. Sure, you can hold on just a sec too. That's fine. So Sean, the lab practical that I received from you had no answers on it.
Okay.
Every question was left blank. So what that said to me was there was probably some technical issue when you tried to submit it. So you are absolutely welcome to take it again.
Okay, so I should just be able to click like the begin and go
Yep.
for it?
Yep,
All right,
you should be
great.
able to go right back in and take it again.
All right, great. Thank you so much.
Okay, you're welcome. Other questions?
Mine is similar to that too. I did mine and I sent it through, but when I went to go check for grades, you know how sometimes it shows you whether you submitted it and it's just not yet graded,
Right,
or because it has that little thing on the side.
right.
Mine did not have that, so I wanted to ask you, do you have mine or no?
What I'll do after class is I'll check and I'll
Okay.
let you know.
Okay, thank you.
You're very welcome.
I also have a quick question, if you
Yes,
don't mind.
no
So I'm
problem.
finishing up my pathogen assignment, and I just want to verify one thing with you.
Yes,
So I'm talking about the MSA, the auger plate.
yes,
And I just wanted to confirm that when it says 7.5 next to it in the PowerPoint, that's the percentage of the salt concentration, right?
that's correct.
Okay,
The
perfect.
7.5 or the 10 is for the salt only.
Okay, perfect. I just wanted to double check that.
Sure.
Thank you.
Who else?
Well, I kind of just sent the message to you privately.
Oh, yeah, yeah. Ashley, why don't I will send you an email after class, okay?
All right, great.
I'll do that right after class. And is there anything I can do for you, Lacey? Anything I can do for you, Lacey? All right. If there is anything I can do for you, send me a message, okay? And we'll take care of it. All right. Have a good day.